Welcome to the Deep Dive. We're here to unpack complex research, giving you the core insights you need. Today, we're getting into biological invasions, and specifically, this really fundamental idea, how simple numbers seem to dictate success.
That's exactly it. We're basing this discussion on a key review paper from 2015. This is by Tim Blackburn, Julie Lockwood, and Philip Cassey, called The Influence of Numbers on Invasion Success. And their core argument, really, is that predicting invasion success, it's not just about looking at species traits or their genes in isolation. You absolutely have to track the sheer quantity of individuals at every step of the way.
I like that. So the mission for us today is really to follow that thread. How does the number of organisms shape everything? Whether they even get transported, if they survive arrival, how fast they spread. It's presented almost like a predictive shortcut, right? To figure out which species flip from being just present to actually, invasive.
Exactly. And what's really neat is how this paper sort of reframes the history. If you go back to the classic works like Baker and Stebbins back in '65, the focus was really heavily on ecological fit or the genetics of colonization, these inherent traits. But Blackburn and his colleagues point out that those early studies kind of missed this central piece, that invasion is often just a series of, numbers games.
It almost seems obvious once you spell it out, but yeah, easy to overlook the sheer logistics behind it all. So, to keep us on track, the paper breaks down the invasion process into 4 clear stages.
Right. First up is transport. That's just getting the species moved beyond its native range, usually thanks to us humans.
Okay.
Second, introduction. This is the actual release into the wild in the new place.
Got it.
Third stage, establishment. This is where they actually manage to form a viable, self-sustaining population. It's not just surviving, it's thriving.
Right, sticking around.
And finally, spread. That's the invasive part, where the population expands its range. And the whole idea is that numbers, quantity, act as the major filter at each of these points.
Okay, let's start right at the beginning then. Transport and introduction. How much does a species' abundance back home matter for whether it even gets moved in the first place?
Oh, it's huge. It's probably the first major sorting mechanism. If a species is common and widespread where it comes from, it's just statistically way more likely to get picked up and moved, whether that's on purpose or completely by accident.
Right, like if you're just randomly sampling cargo or ballast water or whatever, you're going to get more of what's common. It's simple probability.
Exactly. It doesn't matter if it's stowing away in timber or mixed in with agricultural products. The whole process just naturally weeds out the rare species. And we see this even when people are trying to introduce species. Think about birds brought from the UK to New Zealand, the ones that made the journey. Overwhelmingly the common resident species back in the UK.
And there's hard data on this too, isn't there? I remember seeing that analysis on wildfowl.
Yes, absolutely. That study showed pretty clearly that wildfowl species that have been introduced around the world tend to have significantly larger estimated global populations compared to those that haven't.
So the numbers back that up.
They do. The population estimates were just much higher for the introduced ones. It tells us that the very act of moving species, the logistics of it, inherently selects for organisms that are already successful, common, widespread.
So what's the deeper implication there? Is it just that we move common things more, or does this actually pre-select for species that are better invaders anyway?
It really suggests the latter. I mean, if a species is already managing a large, widespread population in its native range, it's often because it's adaptable, it's resilient. It might already be, sort of pre-adapted to deal with disturbed environments, like the human-altered landscapes they often land in.
So the numbers game starts by filtering for potentially tougher competitors.
Exactly. It's not just quantity. It's a kind of quality control imposed by logistics.
Okay, that definitely sets the stage for the next big hurdle, establishment. So a species arrives, maybe in small numbers. Why is that initial phase so risky?
Well, small populations are just incredibly vulnerable. The key idea here is propagule pressure. That's basically the total number of individuals released across all introduction events. And having high propagule pressure is vital because it helps buffer the new population against all sorts of risks that can wipe out small groups.
Okay, let's break down those risks. You hear terms like demographic stochasticity. What does that actually mean on the ground?
Yeah, good question. It sounds technical, but it's basically just random luck in births and deaths. In a big population, if a few individuals die unexpectedly, it doesn't really matter. The average holds. But if you only have, say, 5 birds, and by sheer bad luck, 2 females die before breeding, that could be the end of the whole introduction right there. It's not about the environment being bad, just chance.
Right, pure bad luck. And then you mentioned alley effects. How do numbers play into that?
Alley effects kick in when populations get so small that individuals actually have trouble finding mates, or maybe they can't hunt effectively as a group, or they can't defend against predators. Think of like two moths released in a giant forest. Their chances of even finding each other might be tiny. So high propagule pressure helps overcome these basic social or density-dependent problems.
Makes sense. And we haven't even touched the genetic side yet, the bottlenecks and inbreeding. But before that, how have scientists actually tested this? Does just dumping more individuals always guarantee success?
There's a really elegant study they discuss involving a psyllid insect, Eritidis partiophila, in New Zealand. Researchers did 55 experimental releases with different numbers. They modeled how well the psyllids established based on factors like demographic stochasticity and those alley effects we mentioned. And sure enough, the probability of establishment was strongly linked to the number released. More psyllids, better chance of success.
And this is where it got really interesting, right? They found something surprising at the very high end of release numbers.
Exactly. This was the kicker. They found that the success per individual actually went down when they released huge numbers.
So adding more didn't give proportionally better results after a certain point. Why?
The conclusion was that at those really high densities, the limiting factor wasn't the number of psyllids anymore. It wasn't a lack of mates or random demographic issues. It was the environment itself. Some sites were just fundamentally unsuitable. Didn't matter how many you threw at it. So propagule pressure is crucial, yes, but it can't make up for a bad location.
That's fantastic nuance. And it feeds right into this ongoing debate about the best strategy for release, doesn't it? If you have, say, 1,000 individuals to release, is it better to do one big dump or spread them out over multiple smaller releases?
Yeah, there's been disagreement on that. Some earlier work suggested that if the environment is really patchy or varies a lot year to year, maybe multiple small releases are safer. You're kind of hedging your bets.
Spreading the risk.
Right. But then more recent modeling work, like by Cassey and colleagues mentioned in the review, actually found the opposite tended to be true in their simulations. A single large release was often more successful.
Why would putting all your eggs in one basket be better? Seems counterintuitive.
Their reasoning was that a single large release allows the population to grow much faster right at the beginning. It pushes the numbers up quickly past that really vulnerable small population stage where demographic stochasticity and alley effects are most dangerous. It gets the population established and robust faster, making it better able to withstand any later challenges.
Okay, so speed matters. Now, let's circle back to the genetic side of establishment. Small founder groups mean genetic bottlenecks, right? Less diversity, more inbreeding. How big a deal is that?
It's a very big deal, though it's often hard to untangle from the purely demographic effects we just talked about. When you have a bottleneck, you lose a chunk of the genetic diversity from the original source population. Just by chance, certain gene versions might not make it over. And that loss can really hamper the population's ability to adapt down the line. Less raw material for evolution to work with.
But then you see findings that populations started from multiple introductions often maintain more genetic diversity. How does that work? Is that the genetic rescue idea?
Exactly. If you have individuals arriving from different source populations, maybe at different times, they can bring in different sets of genes. This mixing, or admixture, can replenish the diversity lost in the initial bottleneck. It's like getting a genetic top-up. The paper mentions a cool water strider experiment by Allroth and colleagues. They showed that establishment was more successful if the founders came from two different source populations versus just one, even with the same total number of insects. That mix provided a real advantage.
So diversity matters, and multiple introductions can help maintain it, partly because it often means higher total numbers over time anyway.
Precisely. It all ties back together.
All right, so let's say our invaders have overcome the establishment hurdle. They're here, they're reproducing. What happens next during the spread phase? How do those initial numbers continue to influence things long term?
Well, spread is obviously driven partly by just how many offspring they produce, their fecundity.
Hmm?
More babies equals faster spread, generally. But the less obvious factor is the lasting impact, the sort of genetic shadow cast by the initial propagule pressure.
Ah, yeah, I remember the example they used, the alien birds in New Zealand. That finding was pretty striking, showing effects lasting, what, a century?
Yeah, it's incredible. Brisky and McIntosh studied bird populations introduced over 100 years earlier, and they found that populations started with fewer than 100 individuals still showed higher rates of hatching failure compared to their original native populations. That suggests lower fitness. probably due to lingering effects of inbreeding from that initial small founder size. The genetic scar from low numbers hadn't fully healed even after a century of population growth.
Wow. And does this connection between low initial numbers, low genetic variation, help explain that lag phase we often see with invasions, where a species just sort of sits there for years, sometimes decades, before suddenly exploding?
Absolutely. A long lag phase can often be linked to low initial genetic variation. The population needs time to adapt to the new environment. But if it doesn't have much genetic diversity to draw on, it struggles to evolve effectively. It lacks the raw material for evolutionary rescue, that rapid adaptation process. So it grows slowly, maybe waits for a lucky mutation, or perhaps another introduction event brings in fresh genes and finally kicks off the spread.
And then, assuming the species does eventually spread and become successful, there's this interesting pattern in genetic diversity over even longer timescales, this U-shaped curve.
Yeah, that's a fascinating finding, often seen in established invasive populations. Think about that timeline. Initially, when the population is founded by just a few individuals, genetic diversity crashes. That's the downslope of the U. You get genetic drift, strong selection pressures acting on this small, isolated group. This might go on for, say, 80 or 100 years.
Okay, so the bottom of the U represents the severity of that initial bottleneck.
Exactly. But then, as the population grows really large, starts spreading, and crucially starts to connect and mix with other populations established from potentially different introduction events. You get admixture, genes start flowing between these previously separate groups, and genetic diversity begins to climb back up again. That's the upslope of the U.
And the key point from the paper is that how deep that U goes, how much diversity is lost initially, is largely set by that starting propagule pressure and how much diversity was captured from the native range in the first place.
Precisely. Fewer founders, less initial diversity means a much deeper, more severe drop in that U shape.
This whole review really hammers home how much invasion biology comes down to. Well, logistics, human activity, movement, and just sheer quantity.
It really does. That's the unifying theme. Numbers filter who gets transported. Numbers buffer against bad luck and genetic problems during establishment. And those initial numbers leave a long genetic legacy that influences spread and adaptation potential.
So looking ahead, based on this review, where do your researchers need to focus next? What are the big gaps the authors identified?
They pointed to three main areas for future work. First, really forging a stronger link between the dynamics of transport and introduction, like how many arrived, when, where, from which source populations, and the resulting genetic makeup of the established populations.
Yeah.
We have lots of data on introductions and lots of genetic data, but connecting the two to really understand the journey's genetic legacy needs more work.
Okay, connect the journey to the genes. What's
Yeah.
second?
Second is doing more work, both empirical tests and modeling, to really tease apart the combined effects of genetics, demography, and the environment during establishment, especially at the extreme ends, very low numbers or very high numbers. For example, how much does genetic diversity itself boost population growth, separate from just having more individuals? It's tricky to isolate those factors in real-world invasions.
Disentangling those interwoven factors. And the third gap.
The third is about the long game, spread. We need to better understand the direct link between the amount of standing genetic variation, which, again, is largely set by those founder numbers, and a species' actual ability to adapt, evolve, and spread across potentially diverse landscapes later on.
Understanding the evolutionary fuel for becoming widespread.
Exactly. If we could predict that adaptive potential based on initial conditions, it could really help target management and biosecurity efforts much more effectively.
It's such a compelling case for looking beyond just the inherent traits of a species and focusing on the mechanics of quantity. This whole deep dive really shows how invasion success is shaped, maybe even constrained, by human logistics from the very start. It really is a numbers game.
It absolutely is. And maybe the final thought for you listening is to consider this. Nearly every invasive species causing major ecological issues today first had to win this numbers game. It had to overcome the odds of transport and establishment, often just through sheer volume, whether we planned it that way or not. Our actions set the stage.
A really powerful reminder of how quantity can fundamentally shape ecological destiny. Thanks so much for walking us through the intricate numbers game of biological invasions.